are the ones that grow within the cell wall, between the cell membrane and the cell wall of the plant cells. These are ones that grow in the spaces between the cells of, say, like the cortex of the root. They don't grow past the Casparian strip. They don't grow into the stele or the vascular cylinder in the middle of the root. They stay in the cells on the outside. But what that means is that they effectively increase the surface area that's available for things to move into the plant. So these are our symbionts with forest trees. And in fact, we think a lot of the movement of plants from aquatic systems to land systems were coupled with these interactions with fungi. So there's at least 5,000 known fungi that are interacting, but probably a lot more as we're utilizing molecular tools. The next group, and this is what is actually most of the fungus that is around, but probably one that you're not familiar with, are the Ascomycota. The Ascomycota are what we call cup fungi. They're usually small. Their fruiting bodies are tiny. They, for the most part, are not large fruiting bodies that you see like most other mushrooms, as Basidiomycota. But some of the really important things that are that stuff that get us sick, like molds, things like yeasts that are used in making bread, truffles, the most expensive mushroom out there, morels. Morel season is just about upon us. Everybody hunting for morels yet? What's that? Should be in the next few weeks. As soon as we get a few warm snaps, it's time to look for morels. Yeah. All right. So, some of the really serious plant pathogens that have happened in the United States are Ascomycota. Has anybody here ever seen an American chestnut tree? No. Which is pretty amazing, because 100 years ago, or actually a little more than 100 years ago, 110 years ago, if you were to look in the forests just to the east of here, about 90% of the trees were American chestnuts. Eastern forests in the United States were chestnut forests. And now when you go and look at those forests, what do you see? Oaks, maples, those are the main trees in the forest. Chestnuts were the main tree, 90% of the trees. In 1909, a Ascomycota fungi was introduced from Europe accidentally that basically killed off most of the trees in the United States in a matter of 3 or 4 years. It's called the chestnut blight. And I've seen some surviving American chestnuts. They are coming up from suckers in northern New Jersey. Each year, the blight kills the upper growth, and then the next spring they grow up new growth that then gets killed at the end of the year from the blight. There was just a tree in the last couple of months that a group of researchers in Kentucky have taken the single gene, they've identified the single gene from the horse chestnut, the European chestnut, that makes it not be susceptible to the chestnut blight. And they've genetically modified an American chestnut by adding in that gene, and it's now able to withstand the chestnut blight. So we might get new American chestnuts back, genetically modified American chestnuts that are able to withstand the chestnut blight because of the single gene that blocks one of the processes of that fungi. So also included in this is penicillin. You guys have seen greenish mold on your bread. That's in this group as well. The ascus is this cup-like structure. It's actually on the inside of this. You'll see it even better. These are microscopic cup-like structures. The morel here is also in this group. The location where karyogamy takes place in this group is called the ascus. So with the Basidiomycota, it's the basidium. You see how this works. And in the Ascomycota, it's the ascus. In the ascus, it's the same idea, right? There is a location where the N plus N nuclei come together, fuse to form a zygote in that one cell, and then go through meiosis in the Ascomycota, it goes through a round of meiosis and then an additional round of mitosis. So in the Basidiomycota, we have a round of meiosis. What do we end up with? 4 spores. In the Ascomycota, there's an additional mitosis after the meiosis step. How many spores should we get then?
8.
8. There should be 8 haploid spores. So here's our ascus. Along the inside of this thing is where here's our cup where the ascus is. We're going to start with the mushroomy looking thing, even though it's usually very small. The ascus has the location of those 2 nuclei forming 8 ascospores. The ascospores germinate and form into a primary hyphae. When they fuse and meet together, they actually form little structures that we call antheridium and archegonium, actually ascogonium. And that's the location where those 2 things fuse. Unlike Basidiomycota, where the cells just sort of come together and form this new set, these guys form this fertilization step where the N plus N then grows out of the remnants of the ascogonium. That new secondary hyphae is what forms the mushroom. And that's an N plus N structure. So, okay, so some of the more important groups of Ascomycota include things like yeasts. Yeasts are remarkable for what humans have managed to get them to do. They are linked very closely with humans' use of agriculture because what we were able to do with grains, right? Think about agriculture and humanity is only about 12,000 years old, which is incredibly recent in our history. 20,000 years ago, there were people that looked just like us. Those people probably could have learned calculus. There's nothing about their brains that was less than us 20,000 years ago. But they were all hunter-gatherers. They weren't gardeners. They weren't agriculture. They didn't live sedentary lives growing things. Being able to grow stuff makes it so that you can store that material and eat it over the course of a year. And in fact, replant some stuff the next year. But if you've ever just like tried to take a scoop of flour and eat it, has anybody ever just tried to eat a scoop of flour? It pretty much sucks. Or has anybody ever had like porridge where you just take flour and mix it with water and eat it? I know, it sounds pretty gross, but that's what early people who were saving grains were doing. They were just grinding that stuff up and eating the ground up grains with a little bit of water. Not very tasty. But somebody along the way discovered that if you took that water and flour mixture and allowed yeast to colonize it, the yeast would, through their own feeding process, release a gas that would make that porridge rise up and get a whole bunch of little air pockets inside of it. And then if you warmed that up, it was actually a lot easier to eat. And it was actually a lot easier to store. You could make a bread and it could be utilized weeks later if you kept it in the right conditions. And there's a tremendous innovation and technological advancement for humanity. And the reason why we were able to do that is because we figured out that yeast could be added in to create this fluffy structure. So we use it as an outcome because it's got this ability to make gas, at least for bread. Another side effect, though, of that is that during fermentation, when it's going through its metabolic process without having oxygen around, not only does it put out CO2, it also puts out ethanol. In bread, it's no big deal that ethanol gets burned away, but we also figured out how to keep that ethanol behind, and that's what brought about alcoholic beverages. So our history with yeast is long, but it's linked very closely to agriculture. It's also a damn good model for biology. We use yeast as a biological model because they share so many of the same genes as animals do. So many of the same things that you do on a cellular level, yeast do too. And so you can actually use them as a model for cellular processes in all animals. They're that closely related to us as fungi. It is the first eukaryote that had its entire genome sequenced, and it is the basis of an incredible amount of molecular manipulation. There are vats of yeast that are making drugs that have had genes put into them that make a particular protein that we use as a drug, and it's being grown in a big vat. And the output of this thing is a drug. We're actually, anybody gonna end up in 200 next year? Anyone taking 200 next fall? We're totally changing 200. It's gonna be a totally different course next year. And the lab is gonna be based on a series of experiments that are done with yeast. We're gonna do a bunch of yeast manipulations. Very excited about learning how to use yeast as a biological model. Yeast are single cellular and they grow by these big buds. This is like the formation of a new yeast cell that's going to bud off the other side. So we didn't really talk about fungi in any of the ecological stuff that we discussed, did we? We sort of said, okay, there's decomposers and left it at that. But if it weren't for fungi, if we didn't have this and bacteria, if we didn't have these things out there breaking down dead and dying material and recycling it through their own metabolism and breaking it into small enough components that other things could use, we would literally be buried in lignin and cellulose and other structures that, other biological molecules that otherwise don't get broken down. Fungi helps recycle biological material back to other organisms. That means release of things like nitrogen, phosphorus, carbon compounds. Those are food and nutrients for plants and animals. So fungi is an incredibly important recycler of biological molecules. They also form lots of symbioses. We've talked about symbioses with plants, but there are fungal symbioses with other things too. Some of those symbioses can be obligate. That's things like arbuscular mycorrhiza, the Glomeria mycota, cannot survive without it, without those interactions with plants. Other ones are facultative. They can survive on their own, but can form these associations. They're also important because we've got things like pathogens. There are human diseases that are caused by fungi. They're also important because they may exist as parasites. They may exist commensal. They may exist mutualistic. All the kinds of ecological interactions we talked about in the first unit, fungi are in those interactions. There's even carnivorous fungi. I was just reading about a group of rotifers, the Delonid rotifers, that ingest a fungal spore, and then that spore germinates inside of their gut, and the fungi proceeds to eat out the inside of the rotifer, like dissolves it from the inside out. And then once it's totally dissolved the rotifer, it puts out these little fruiting bodies and sends spores out to be eaten by another rotifer. Okay, so there's some really interesting interactions with fungi that actually are beneficial to plants. Fungi infect plants all the time. So we've looked at ectomycorrhizal fungi. And we've looked at things like smuts and rusts that affect plants and damage those plants, and blights that damage those plants. But there is a lot of research that argues that fungal infections can be helpful to plants. Plants put out, or fungi put out an incredibly interesting milieu of toxic chemicals. We call them mycotoxins. Things that does anybody have a mold allergy? What you're allergic to is the mycotoxin. Anybody ever heard of black mold? Black mold, and some of the toxins made by fungi are some of the most toxic natural things that we know of. Like the black mold mycotoxin is incredibly toxic. In incredibly minute quantities, people can have serious health problems from being exposed to it. But if you're a fungus and we also actually have lots of things that we use that are fungal derived as well. We have our cells have specific transporters that carry fungal derived chemicals into our cells. Fungi make a lot of really interesting antioxidants that act to limit oxidative stress within our cells, and we have cellular surface transporters that bring those fungal-based antioxidants into our bodies. So they're really, really interesting. Anyway, so there are some really cool experiments that have been looked at with herbivory, whether or not a plant is infected or not. So in this particular set of experiments, they showed that when you have a fungal infection, so this is showing, the experiment shows the plant is either infected with a fungi or not. So these are endophytes. They grow within the spaces between the cells. They grow into the leaves and into the spaces between the cells. If you have a fungal endophyte, herbivory is reduced compared to if you don't. So think about plants that are being preyed upon, right? That have herbivory. We talked about plants making their own chemicals as a way to limit herbivory. There are some plants that make chemicals that actively induce infection from fungi. Chemical signals, flavonoids and things that fungi recognize as being as an attractant and will grow to the places where those are. And so plants can actually influence where fungi grows within their leaf structures based on the secretion of these flavonoids and increase the infection rates in parts of the plants that are most likely to be preyed upon and can reduce herbivory through an interaction with their endophytic fungi. There was a guy at Rutgers who I saw give tons of talks on this. He was an Amish guy, literally had an Amish beard and stuff, who studied fungal associations with plants. He showed that even things like grazing by cattle and sheep can be influenced by fungal endophytes. That sheep and cattle will not graze as much upon grasses that have been infected with certain endophytes. So pretty cool that the toxins that the fungi are creating to save themselves are actually helping the plants that they are invading. There are also some very tight associations that are obligate mutualisms that we've essentially started to consider as a species, as their own species, and those are lichens. Lichens are an association between a fungus and a photosynthetic partner of some sort. Usually the photosynthetic partner is an algae, but sometimes they are cyanobacteria, or even in some places some other strange photosynthetic protists. So what we end up having is a fungal organism that most of these are Ascomycota. So they have shapes that look very Ascomycota-like when you look at them. They'll have ascus, they'll have spores being made in an ascus. But when you look closely at the structures, you'll see algal cells embedded within the fungal cells. And what's happening is that the algal cells are photosynthesizing, and that's giving the fungi photosynthate. But the fungi is protecting the algae from often extremely stressful conditions. And so lichens end up growing in really remarkable spots. Go outside after class and look at any tree out there, and you will see lichens growing on it. They're either crusting along the surface or lifting up, or there's even some that are you ever seen Spanish moss hanging from the trees in the southern parts of the United States? That's actually a lichen. It's not a moss at all. So lichens are beautiful. I have told you about my friend who has like discovered the genetic signatures of hundreds of mushrooms, right? He had this coffee table book at his apartment that was photographs of lichens. And they come in all kinds of interesting colors because of their algal symbionts. And the book was a science book. It was called Lichens of the World. It had about 2,000 lichen pictures in it. And I just sit there and look at these beautiful photographs because there is so much diversity in their colors and their forms and their shapes. They're often the first thing that's found in brand new locations that are. So when we talked about succession, often the first biological organism that shows up is a lichen because they can get what they need from their algal symbionts, and that means they can live on bare rock. So you can go out and see them growing on sidewalks, like straight on concrete. They're remarkable organisms. So anyway, even though they're really able to handle harsh conditions, they're also highly susceptible to air pollution. And they're often used as a proxy for determining air pollution and air quality. One of my friends who worked on lichens for a summer in Alaska showed that the diversity of lichens as you move in towards Anchorage diminishes quickly as you get towards the city because of pollutants from automobiles and from people burning oil to keep their houses warm. Those particulate pollutants really damage the lichens. And so even just 20 miles away, 15 miles away, the diversity of lichens outside of Alaska or outside of the cities rises a lot. So they're actually used as a proxy for understanding levels of air pollution when you can't permanently monitor all the time what's in the air. So we talked a bit about mycorrhizal. These are mutualistic relationships between fungi and plants. They're incredibly common. Most plants go through some kinds of fungal associations. They act as extensions of the root system. So here we're looking at the root. But all of this white around the root, that's all hyphae. That's all fungus. And so essentially it's just upping the amount of surface area available to the plant for absorbing nutrients. We've talked about our muscular and ectomycorrhizal, mostly Glomeromycota, mostly Basidiomycota. There are some really cool animal symbioses. Has anybody ever seen leafcutter ants? Anybody ever been to the tropics? Leafcutter ants are really common in the tropics, and what you'll usually see is like a little pathway that you can see is actually like beaten down, and a line of ants with each ant carrying a little leaf on its shoulder. And we used to think that they were actually eating the leaves, but it turns out that leafcutter ants are fungal farmers. They're cutting off the leaves of the trees and moving them into their underground burrows and inoculating them with a fungus that then grows on those little leaf parts, but the ants eat the fungus. And as we've gotten to use molecular tools, it turns out that they have obligate symbioses with fungi that cannot exist outside of the ants anymore. There are ant species-specific fungal strains, and when a new queen goes off to establish her new nest, she takes a little bit of the fungus with her. She takes her own little fungal patch to inoculate the new set of fungi, or the new set of leaves that her workers are going to go collect for her. That's how close the association is. The association is also, or mutualistic associations are also so important that it helps make it so that animals that eat plants are able to break down that cellulose. So if you think about things like cows and stuff, they eat grass. But much of the grass that they eat, much of the energy they obtain from the grass is in the cellulose that they eat. So how do they get any energy out of the cellulose? You and no other animal has the enzymes to be able to break those cellulose down into the component monomers. Essentially, it is, hey, you ever eat, you know when they say fiber? They're talking about cellulose. Fiber, for you, is something that you cannot actually digest. And it bulks up in your gut, and you expel it basically unaltered. You know what they're now using is cellulose? If you ever see cellulose in food product, like labels, if it says cellulose, do you know what they're using to do that? Mostly sawdust. It's now an additive with food because you're allowed to add sawdust in food. It's just called cellulose. All right, so those organisms that eat plants exclusively cannot break it down themselves. And so they actually have symbionts, fungal symbionts within their guts that are able to break down the cellulose. So in things like cows and deer and other ruminants, they often have a fungi that is of the Neocalamanthagoda group in their guts that's what's breaking down the cellulose. So there are lots of diseases caused by fungi, lots of plant diseases, things like smuts and rust. Some of them are incredibly harmful and damaging to agricultural crops. Some of them are damaging to agricultural crops, but people eat them. It's sort of gross. Food that can be spoiled by fungi. Who's ever seen like gray mold in their applesauce after leaving it in the fridge too long? Chuck that out, right? Mold on their bread, chuck that out. Those are all fungi that are influencing humans and human use of things. Anybody ever eat this? What's that? It's a delicacy. It looks real gross, but it's a fungus that's invaded individual corn kernels and swells up. But people like to eat it. There's all kinds of crazy toxins made by fungi. There are things like Fusarium, which is a wilt that wilts. It's a common agricultural pest. You get Fusarium wilt. It actually makes a toxin called vomitoxin. What do you think that does? It induces expelling bacteria. Yeah. So so I've had Fusarium wilt in my garden, and you don't want to eat that lettuce.
Like
Is that what?
Like Apicac.
Apigac?
Apicac. So like little lazy frog.
It's like what they give you if you.
I don't
It's
know.
a liquid.
It's a liquid. Maybe it's the same thing, or at least a similar compound. Aflatoxin causes anaphylactic shock. This is asparagus. This is a really common fungi in your food. Some of those mycotoxins are so incredibly toxic that really small amounts cause health issues. So fungus can have that's a pretty bad case of athlete's foot, right? If you've ever had yellowing nails, that's a fungus. If you've ever had a little brown patch on your skin, those are often funguses. Fungus invade humans all the time. Hard to get rid of too. So ringworm is actually just another name for the same fungus that gives you athlete's foot. It's also jock itch, same fungus, just on a different part of your body. This is one that actually happens very often with nursing babies, called thrush. This actually can get infect the nipple area, and then you just pass it back and forth between you and your baby for like weeks and weeks and weeks. It's really kind of tough because you can't put a topical cream on it and still continue to breastfeed your baby. So oftentimes really bad thrush infections will make it so that in the past, we just kept breastfeeding through the whole thing, but now you'll often shift to formula because of it. Yeast infections, fungal infections, so vaginal infections. We are invaded by these things all the time. So the take home is, fungi, well, probably up till this point in your life has been considered boring and not very exciting, unless of course you have mushroom logs and aphilated that in your backyard and you eat them. They're actually pretty important. They are incredibly diverse. They are important in the recycling of natural products in nature. They've got their own weird way of cell dividing. They're also interesting in their symbiosis with other organisms and in their pathogens for stuff like that. And if you want a career in science, this is a field in which the doors are wide open. A couple of years ago, we put out a call for, we were needing to hire a new geneticist, a second geneticist, for our bio department. And we put out a call for we wanted a fungal geneticist. We had 180 applications. Do you know how many of them were actually fungal geneticists? 0. There are not many fungal geneticists out there, even though it's an incredibly interesting group of organisms. Sort of funny, it was like we recognized that this was a deficit in our curriculum. We wanted to get a biologist that could plug it, and there was no biologist that could plug it. So we hired a fly geneticist. Dr. McClure, he left last year. She was here for 3 years. All right, fungi, awesome. That's the take home. Everybody take 2 deep breaths. Any questions about fungi before we move on to human anatomy and physiology? Just kidding. No? No questions about fungi? Which means you're all ready for the thing you've all been waiting for. All right, what are we going to talk about now? Animals! Awesome. Wait, what the hell? Are those things even animals? There's only one thing up there with a face. So, I do this thing in my FYS. Anybody here was in my FYS? No, I do this thing in my FYS. So my FYS is called Understanding Animal Diversity. And on the first day of my FYS, I ask, what's your favorite animal? Okay, and so somebody scream out their favorite animal. Lion. Tiger. Lions are always big cats. What? Did you say lion? Yeah. Lion, tiger. What? Dogs, elephants. Come on, keep going. Dolphins, koala. Otter. Manatee. Pigs. Cows. Snakes. Ooh, the first one that is not a mammal, but yet still a vertebrate. Sea turtles. Turtles. What? Manta rays. Also a vertebrate. Every single one of the things that you guys just mentioned is a vertebrate, right? How many of these pictures up here are vertebrates? Just one, a fish. Not one of you mentioned a fish. Every single one of you mentioned a terrestrial mammal, except one which is a terrestrial reptile. Sharks are cool, also a vertebrate. In fact, vertebrates are not even a phylum. If you look at the diversity of life in terms of animals, vertebrates are a subphylum. These are not even a phylum. They're actually a subphylum of a relatively obscure phylum that is not particularly diverse in terms of the number of organisms. So I'll give you an example. How many mammal species are there? You guys all mentioned mammal species. How many are there? How many mammal species are there on the planet? A couple hundred. You could come up with a couple of hundred on the top of your head right now. There's more than a couple of hundred mouse and rats, but actually that group is the largest group of mammals. So how many mammal species are there? Huh? Mammals? It's about 4,500 mammals. About 4,500 mammals. So, but you know what this lets me know is that you guys don't really know anything about animal diversity, right? 4,500 mammals. How many birds? A lot. About twice as many, 10,000 birds, okay? How many amphibians? Not as many, about 2,000. How many reptiles? About the same as the number of mammals, about 5,000. Okay, so we're at 2,500 or 25,000 vertebrates. All the terrestrial vertebrates are 25,000. How many fish are there? More than there are all the others. So about 35,000. So between those two, we're looking at 60,000 species. All of the vertebrates, fish, mammals, everything you can think of, 60,000 species. There are more snail species than there are vertebrates. About 65,000 snail species. Yeah, okay. How many beetle species are there? 10 times that, 600,000 beetle species. In fact, there's this really famous quote where they asked this guy, Theodor Dubinsky, who was a systematist. You know, this was in the 1930s, and they asked him, what has he learned about God from studying the diversity of life? And his response was, God has an inordinate fondness for beetles. Literally, half of the named organisms, or half the named animals on the planet, are beetles. So, I know you guys are like, yes, we're finally talking about animals. We're not going to talk about what you think about as animals for at least 3 weeks. We are not going to talk about vertebrates for weeks. You guys are going to learn all of these other groups before we get there. You're going to learn about some really, really weird, bizarre invertebrates that do cool things. Now, there's about 35 named phyla. These are still sort of, we're not quite sure about some of these phyla. You're not going to have to learn all of these phyla. There's only about 15 in here that we're really going to focus on. Things like the Luricifera. We're not going to talk about those. They're pretty cool. But what we are going to talk about are some of these really big groups and then a few individual phyla out of it. Phyla or phylum is the equivalent to division that we talked about in plants. The big grouping that all share a fundamental underlying body plan. So one of like the biggest phyla, it includes the beetles. What would that be? The arthropods. Those all have an exoskeleton. So things like spiders are arthropods. Insects, arthropods. Crabs, arthropods. We'll talk about those different groups as we go. So, this is the next few weeks of our term. We're going to be able to think about all the diversity of animals that are out there. But in order for us to be able to deal with this incredible diversity, you guys, right, plants are cool. A sporangium is a sporangium is a sporangium. Each one of these animal phyla has their own body plans, has their own setups. Some of them have their own types of the way their structures are put together. We have to come up with a framework to understand this diversity. We have to come up with some way to recognize how to group things together in coherent, sensible ways. So let's talk about what animals actually are. There are some general features that animals share. One, they are heterotrophic. What is that? That's right. They cannot utilize sunlight or, in some senses, chemicals to be able to generate their own carbon compounds. Plants and things like chemosynthetic bacteria can't. Now they can form symbioses with plants and chemosynthetic bacteria, but it's actually those other organisms that are doing the synthesis of carbon compounds. So they have to consume other things to get their energy. All animals are multicellular. They are groups of cells together functioning in a coherent system. So they're heterotrophic and multicellular. None of them have cell walls. That's something that only plants have, and only fungi and a bunch of assorted protists have cell walls as well. They are generally, or they're structured with tissues. Tissues are groups of cells that are acting together in a coherent function. So things like skin is a tissue, groups of cells that are acting together as a coherent function, separating the inside and outside of the organism. They're, for the most part, active, not always, but for the most part they're active. That means they have nerves and muscles that are able to make them move. They're often flexible, although rigidity happens in some groups. So, but movement is something we see very often. Their diversity is mind-blowing. You guys have shown me that your understanding of the animal diversity is limited, right? You think animals, you think things with fur and faces. That is a small diversity in terms of what's out there in animals. More than 99% of all living organisms around right now, that are of all animals that are around right now, are not vertebrates. They're invertebrates. So the sort of vertebrate-invertebrate split that we've made in how we view animals is our human bias, right? We are vertebrates, so somehow we think they're more important or cooler than all of the other stuff out there. I spent my entire biological career avoiding learning anything about vertebrates because I find them immensely boring. I'm a vertebrate. I don't really want to know about myself. I'd much rather know about the weird, bizarre stuff that's out there. So we're going to spend a lot of time talking about the invertebrates that are out there. Their diversity of habitats is huge. They live everywhere, from the bottom of the deepest ocean to the top of the highest mountain to the air around the highest mountain. Animals have managed to invade every habitat on the planet, including some really amazing places. Like, there is a worm called Albanea pompigea that lives in superheated water in underwater volcanoes. Water at like 300 degrees Celsius. Organism lives there, in those waters. That's just crazy talk. It really does. There are organisms that live in ice. There's a set of insects that are called ice bugs. You find them living on the glaciers in Antarctica. They literally live everywhere. Okay, so these guys have no these have sexual reproduction. They have no haploid cells other than gametes. So there is no gametophyte, there's no alternation of generations. All animals are diploid, except for egg and sperm, which are haploid that fuse together to form a diploid zygote that become the next generation. The origin of animals is actually controversial. There were a couple of different possibilities of where multicellular animals came from. Multicellularity has its origin in there were a couple of competing hypotheses. So when I was in college 25 years ago, these were competing ideas. One of them is called the multinucleate hypothesis. Do you guys remember when we looked at chaos way back in 200, the amoeba? We We looked at an amoeba in lab, and the amoeba, if you look close, it had this sort of like amorphous shape, right? And if you look in the amoeba, you'll find that it is what is called multinucleate. So it's a single large cell, but within it are a whole bunch of nuclei. And each nuclei inside of there regulates the expression of proteins right nearby it. So each one of those nuclei is actually making messenger RNA, and that messenger RNA is extending out into the areas around it. The ribosomes are making the proteins. So different nuclei can actually control different levels of protein expression at different parts of the cell. So one idea is that there was a multinucleate protist, and we know they exist, that was able to figure out how to, or evolve to be able to pinch off the insides of the membranes between the cells, or between the nuclei, forming a multicellular organism. That's called the multinucleate hypothesis, that they arose out of a single multinucleate cell, we see it in amoebas, to become multicellular. The other hypothesis that was being thrown around at the time was what we call the colonial hypothesis. And that idea is that groups of protists that have flagella were able to form colonies where when you divide your cell, you don't release the other cell, and they don't ever end up going off and being free living. You just have another cell next to you. And in fact, there are some groups of organisms, in this case, there are colonial choanoflagellates, that form these structures of individual cells attached to each other, forming a sort of ball of cells. What's cool about these guys is that they all have these little collars sticking out the side that have a flagella. And the way they feed is that the flagella beat and move food towards the cell, and then they move that food into each individual cell. That's the colonial hypothesis. There's some argument that maybe both of these things actually happened at early animal development, and that there might be some groups that are derived from one and some groups that are derived from the other. Because we do see some animals that form structures that look like this, that form structures that are like multinucleate tissues that are called syncytial. So this is sometimes called the syncytial theory. All right, so we now have molecular tools. We now have ways to look at the sequences, mostly of ribosomal RNAs, the structure of the sequence of the DNA that makes the ribosome. You guys know ribosomes are incredibly important. If your ribosome gets mutated, it doesn't work. You're not surviving. It's what makes proteins. So the sequence of ribosomes does a very good job of showing relationships between organisms. And now that we have ribosome and ribosomal RNA, when you form a tree, and you guys saw it back when we looked at the last lecture, go back to that eukaryotic, go back and look at the eukaryotic tree, slide number 3 of the start of fungi, and tell me which one of these theories is probably the most likely. Go look for the multinucleate amoebozoids, and go look for the flagellated choanoflagellates. Who is sitting closest to animals on that tree? Are you sure? The choanoflagellata. They're right there, branched right next to the animals, right? Choanoflagellates and animals are together, and just off to the side of that are fungi. Of all of the protists, the most closely related to the animal line are the choanoflagellata, the choanoflagellates. So who is the most, which one of these theories is probably the most reasonable for the origin of animals? The colonial hypothesis. And in fact, when we start looking at sponges, we're going to see the same cell structure. So it argues that that is in fact where our origins are, from choanoflagellate colonial groups. Okay, so how we got multicellularity is one of those possibilities. But in the evolution of animals, we see some very clear themes. These are sort of like key transitions, key ideas that we see that help to make the diversity that we see in animals now possible. Those are the development of tissues, alterations of symmetry, how the interior parts or interior cavities of the animal are structured, how they develop, and the repetition, the serial repetition of body parts that allow for larger and larger organisms. That's called segmentation. So, let's talk about this. When you become multicellular, there are some immediate problems that have to be dealt with. For example, when you're small, every single cell or every single part of your body is close to the exterior. And so by simple diffusion, things like gas moving across the membrane, you can easily get every cell within there. If you had one big giant cell and diffusion happened the same way, you'd end up with this whole part in the middle that just couldn't be supplied by things diffusing across the membrane. So multicellularity may have been able to account for that problem. By having multiple little cells all next to each other, every single cell could still be supplied. So there's a way to get bigger without actually having your cells get bigger. Bigger might have been a competitive advantage to other cells because now you are able to either engulf other cells or you're able to acquire more nutrients because you're actually covering more area. But as you keep getting bigger and bigger, you still have this same problem. The interior cells now become further and further away from the environment. And so getting gases to interior cells become one of the major problems that has to be dealt with. And we'll see over the course of the evolution of animals that there have been a few general ways in which you can do this. You just increase the surface area, and in some cases, you could evolve a system that can bring those gases deeper. So respiration and gas exchange is one of the things that has to be dealt with as you get larger and larger. Also, as you get larger, your need for nutrients increase. And so you have to consume and acquire more nutrients than you would if you were just small. Now think, if you're a small organism and you can just pick things up from the surrounding environment, the larger you get, the faster you have more cells to supply, the faster you're going to deplete the surrounding nutrients. And very quickly, if you can't move, you're stuck with having no food around you. So that getting bigger means that you can now move to other locations to find new food or put yourself in locations where food comes to you. So the need for nutrition goes up, and that means that there's a need for acquiring that energy and moving that energy around the body of the animal to the cells that actually need it. When you're small and you have wastes to get rid of, you just secrete those wastes right over the cell membrane and out it goes and away it goes. But if you're big with multicellular, what happens if you secrete toxic wastes like urea out of a cell and there's another cell right there? You're basically dropping your toxic waste as a cell onto your neighboring cell that's still part of your own organism. So some way to deal with that waste and to get it away from the cells without damaging the cells is something we're going to see as a theme that happens as organisms evolve in multicellularity. Being able to coordinate your movements, individual cells have to be able to talk to other cells. Those cells have to be able to act together in a coherent way to be able to make the whole organism move. So coordinations of movements and activities between cells and between the whole organism itself is a theme we're going to watch happen repeatedly in different groups. What about the need of getting those nutrients deep inside the organism? When you consume another organism or you consume other things, those nutrients are limited in where they actually end up. If you've got a gut, they're in this gut. How do you get that from the gut to the actual cells that need them? How do you get the gases from the places where they're exchanging with the exterior environment to the cells that actually need them? We're going to see developments of circulation systems, ways that you can move those things around the interior body of the organism to get to the cells that actually need it. The last thing that is a major problem is reproduction. When you are a organism, you have to find another organism that is compatible with you, in which you can put your gametes together with them. So when you're in water, and it's not that big of a deal, what do? You just spew your gametes up into the water column and they find each other. But when you're on land, you now have the same problem that we saw in plants. In fact, if you look back at each one of these things, how do you deal with circulation? How do you deal with waste removal? How do you get energy acquisition? How do you have gas exchange? Those were all things that we saw were a problem in land plants, weren't they? Right? Those were all problems that had to be overcome. How did they overcome the respiration gas exchange problem? Stomata and moving air into and out of their body parts. How did they deal with nutrient acquisition? Xylem and phloem, moving things around the interior of the body. That's also like circulation issues, right? How did they deal with reproduction? Seeds, flowers. How did they deal with waste removal? They actually do it by dumping it out and throw it flow, and we didn't talk about that at all. So many of the same problems that plants had to figure out or had to evolve solutions for when they moved from water to land. Animals had to figure that out too. And so as we work through the evolution of animals and the different groups of animals, you need to think about how it's similar to what happens to plants, because the evolution of this is remarkably similar. So